In this lecture, we are going to talk biotechnology, looking at the medical and industrial applications of genetics, which were the subject of our previous lecture. So here's the thing about biotechnology. Biotechnology is a huge, huge venture that involves so many different kinds of genetic applications that really it would be difficult for us to talk about all of them in a single lecture. In fact, you can get a degree in biotechnology. You can take courses that devote themselves to different aspects of biotechnology. So what we're going to do here is try to apply the concepts that we learned in our genetics lecture to certain aspects of biotechnology. And so what we're really going to talk a lot about is using recombinant cells, which, as you remember from the previous lecture, are recombinant because they have DNA inside of them that has been donated to them from an outside source. So that was something we talked about in the previous lecture. We talked about horizontal gene transfer as a natural phenomenon. Today, what we're going to do is talk about horizontal gene transfer as an industrial phenomenon, an in vitro phenomenon, where we are preferentially inserting DNA into recipient cells in order to recombine them in such a way that they can have some therapeutic application. Those applications are very wide-ranging, and they might include the in vitro production of human enzymes or other proteins. They might include the production of subunit vaccines or the experimental viral DNA vaccines that you might be hearing about because of the COVID vaccine. They include gene therapy applications, disease identification applications, and they all have in common that at their heart there is a gene of interest, as it's called, that has been cloned or copied from a donor organism and then is placed into a recipient cell in order to express that piece of DNA. And so what we call all of that together is recombinant DNA technology. Recombinant DNA technology is the use of in vitro produced recombinant cells in order to make some therapeutic or industrial product for widespread use. If you're interested in some of the pharmaceutical products that are produced as a result of recombinant DNA technology, there's a table in the lecture notes that you can access. What we're looking at here is just a very basic overview of this process of creating recombinant cells. What we're going to do after we look at this very quick overview is go and look at each of the constituent parts and see what microbes have played a role not only in producing these pharmaceutical or industrial products, but in making the cells that are able to produce these pharmaceutical or industrial products. So it begins with, in this case, a human cell which has a gene of interest that is insulin. This is an incredibly important pharmaceutical product throughout the entire world for all people who have insulin-dependent diabetes. And prior to the recombinant microorganisms making insulin, it was necessary for insulin to be harvested from the pancreas of slaughtered pigs, which led to the possibility of zoonotic or animal transferred diseases. It also produced an insulin that was not quantified as carefully. And so this revolution in the ability to create, so this revolution in the treatment of insulin-dependent diabetes, which begins with bacteria or yeast cells producing insulin, has really revolutionized the way that diabetics can be treated and has done a ton to increase both the longevity and the quality of the lives of insulin-dependent diabetics. So insulin here is our gene of interest. Insulin is the thing that we want microorganisms to make for us. So we need to cut that gene out of the human genome and we need to insert that DNA into a bacterial cell. Now, we know that there's a process of transformation, and transformation can occur when a piece of DNA is found in solution and taken up by bacteria. That's a very uncommon process, and as you might remember from our previous lecture, it relies on the bacterial cell being able to use and transcribe and translate that protein, and it also is necessary that that cell is able to pass on that protein to its offspring. And the easiest way for that to happen is for that gene of interest to go into the cell as a plasmid, because a plasmid is a self-contained piece of DNA which can be transcribed and translated into proteins and can also be replicated. So you've got to find a way to get your gene of interest into a plasmid, and then you've got to take that recombinant DNA plasmid, right? That plasmid itself is recombined because it has the plasmid DNA shown here in blue and the human DNA shown here in red. And then you have to get that DNA into the bacterial cell, creating that recombinant cell, which will then transcribe and translate the human insulin. And then you've got to collect that insulin in enough quantity that can actually be commercially useful to you. So that's the overall process. The question is, how do we make this happen? And the first issue that we have is, how do we cut a gene out of a chromosome? How do we get just a single piece of DNA out of a chromosome? And then how do we cut a plasmid such that we can get that other piece of DNA into the plasmid, creating the recombinant DNA molecule? So what you need is a really, really small pair of scissors. In 1976, those little tiny molecular scissors were discovered, and they were given the name restriction enzymes. And they were named restriction enzymes or restriction endonucleases because they could restrict or cut a nucleic acid. And they were discovered in bacteria, and they actually comprise part of the bacterial defense system. It's sort of like a bacterial immune system where if invading viral DNA from a bacteriophage enters the cell, these restriction enzymes will become activated and they will go chop up the viral DNA. And if the viral DNA is chopped up, then it can't direct the synthesis of new viruses. And so the bacterial cell is safe from the end result of viral infection, which is lysis of the cell. What researchers discovered was that those molecular scissors could be used to cut or restrict a piece of DNA in vitro in the lab. So you see here a table with 4 different restriction enzymes. I'm going to point out just the second one and we'll focus on him for a while, and that is a restriction enzyme that was discovered in Escherichia coli and was given the name EcoR1. Notice eco in italics to indicate to you that that's from its genus and species name, and R1, a designation indicating that this was the first restriction enzyme discovered in E. coli. And these restriction enzymes don't just cut DNA at any site. They actually have what they call a recognition sequence, which for Escherichia coli is this palindromic sequence, GAATTC. And when EcoR1, the restriction enzyme, is allowed to digest DNA, it doesn't just go through and cut the piece of DNA inadvertently. It attaches to the DNA molecule and it travels along the DNA molecule until it finds this recognition site, and it cuts the sugar phosphate backbone between the A and G nucleotides, which destabilizes the molecule and causes the hydrogen bonds between the A's and the T's to break and opens up the DNA molecule. So these restriction enzymes are said to digest DNA, and when they digest DNA, what we mean by that is that they are moving physically along a piece of DNA, and everywhere they find this restriction sequence or restriction site, they cut the sugar phosphate backbone, thereby opening up the piece of DNA. And the really cool thing about EcoR1 and a couple of these other enzymes is that when they cut the sugar phosphate backbone of the DNA, they create these single-stranded fragments, which, for instance, with EcoR1 is this AATT. And that's cool because that AATT single-stranded piece of DNA will be drawn to and want to attach to any complementary sequence, because that A will want to bind to a T, and the A next to it will want to bind to a T, and the T next to that will want to bind next to an A, as will that final T. And because that AATT single-stranded sequence wants to bind its complement, it's called sticky. And that stickiness of the DNA is what enables us to combine molecules of DNA from two sources. And that's a process that takes a little bit to understand, so we're going to take a moment to look at what that process looks like in vitro.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as EcoR1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of EcoR1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, EcoR1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
Okay. All right. In this particular example, you have a restriction site on the plasmid GGATCC, which is the restriction site not of EcoR1 but of BAMH1. So this is a different restriction enzyme, and this restriction enzyme, regardless of the site, still has the ability to create those sticky ends because it breaks the sugar phosphate backbone between those adjacent guanine nucleotides. So when you digest this plasmid with BAMH1, you will create a linear piece of DNA which has these sticky ends. So in a separate test tube, you take the cellular DNA, right? In this case, it would have been the human genome, and you use the same restriction enzyme to digest the cellular DNA, and then you isolate out the piece that has the insulin gene within it. So now that little red piece of DNA, the portion that is not read out is the insulin gene. So now you have your insulin gene isolated, and you have in a separate test tube your plasmid, which has been opened up, so now it's sticky and wants to attach to that. And when you put those two things together, they can anneal to one another, they can attach to one another, they can attach at those sticky ends, because the GATC portion of the human DNA is exactly the same and is complementary to the CTAG portion of the plasmid. So not in all of the plasmids, but in some of the plasmids, you get this recombinant DNA. You get a piece of DNA that has that insulin protein within it, that has the gene of interest. Now the question is, how do we get it into a cell, and how do we recognize that that cell actually has the gene of interest in it? Because we don't want to just assume and allow all of the bacteria that we've exposed to this recombinant plasmid, that all of them have been recombined. We need a way to select for the recombined cells. Notice that on this plasmid, in addition to that site where that gene of interest is taken in, you have an ampicillin resistance gene. And that ampicillin resistance gene is going to allow us to select for bacteria that have been transformed, because all transformed bacteria, in addition to being able to make insulin, will also be resistant to the antibiotic ampicillin. So all you have to do is grow them in the presence of ampicillin. So you take this vial that contains a couple of different things. It contains chromosomal DNA that's floating around by itself. It contains plasmids that have no chromosomal DNA inside them, and it contains some plasmids that have the chromosomal DNA inside them. So your first step is to transform in vitro bacterial cells like E. coli with this plasmid. So some of the bacteria will not pick up any plasmid at all, and because they haven't picked up any plasmid at all, you can select against them or kill them by putting ampicillin in your petri dish, right? And because they're not ampicillin resistant, they won't survive, so you will have selected against them. The other two possibilities are that the bacterium takes up the plasmid, or the bacterium takes up the plasmid that contains the insert, the gene of interest. Both of those will grow on media containing ampicillin because both of them have the plasmid. So now you need to ask the question, how can I differentiate between the bacteria that have the plasmid and the bacteria that don't? I want to talk just for a moment and then actually show you another video where we talk about differentiating between those two cells that actually are recombinant in order to figure out whether or not those cells have the gene of interest. And that has to do with the fact that the area of the plasmid where the restriction recognition site exists in what's called a multiple cloning site, where there are multiple restriction enzyme recognition sites within the beta-galactosidase gene. You might remember that beta-galactosidase is involved in the catabolism of lactose. It's part of the lac operon, and it codes particularly for an enzyme that breaks down lactose into glucose and galactose. So when this enzyme is present, the cell will be able to break down lactose. But what makes this useful in vitro is that beta-galactosidase can also cut apart a molecule called XGAL and turn it blue. When the cells are grown, if the beta-galactosidase enzyme is functional, you can add a colorless substrate to the medium and the cells will turn it blue. When this happens, what that says is that plasmid has opened up because of the use of the restriction enzyme, but it has gone back and re-annealed to itself without taking up the gene of interest. So the blue colonies are actually those that you do not want. What you want is a cell that has opened up its plasmid, taken up the gene of interest, and disrupted the beta-galactosidase gene so that it cannot make the beta-galactosidase enzyme and cannot change the color. So the white colonies are those that we can select for based on this phenotype that have the gene of interest, as evidenced by the fact that they cannot convert the XGAL to a colored product. So again, I think this is something that makes a little more sense when we see it happening dynamically. So I'm going to show you another quick video.
Plasmids can be good cloning vectors because they carry an origin of replication and are therefore able to replicate independently within a cell. Most plasmids used as vectors also encode some type of selectable marker, such as the gene for resistance to ampicillin. If the host cells are ampicillin sensitive, the only host cells that can grow on a medium containing ampicillin are those that have taken up the plasmid. Vectors must also have a small sequence of base pairs that can be recognized by a restriction enzyme. When this enzyme opens the circular plasmid, foreign DNA can be incorporated. When the plasmid vector and foreign DNA are both cut with the same restriction enzyme and mixed together, not all molecules will join to form recombinants. Some vector molecules will reanneal without incorporating foreign DNA. To identify cells that contain plasmids that have incorporated foreign DNA, a second marker gene is needed on the vector. This second marker contains the restriction enzyme site within its nucleotide sequence. If foreign DNA is inserted, the second marker is inactivated. This is referred to as insertional inactivation. A common second marker is the LAC-Z gene, which codes for the enzyme beta-galactosidase. Beta-galactosidase can cleave a colorless chemical called XGAL to form a blue compound. Therefore, colonies of cells that harbor the intact vector but no new recombinant DNA can make beta-galactosidase and form a blue color in the presence of XGAL. However, colonies that contain new recombinant DNA cannot make beta-galactosidase and are white.
All right, hopefully having seen that happen dynamically, you have a little better idea of how that process actually works in the cell and in the lab. What I wanted to do before we move on from this concept is just relate what we're learning here in lecture to a lab that we're actually going to be looking at as well, where we are undergoing an in vitro transformation, not with this plasmid we've been talking about, which is the PUC19 plasmid, but with a plasmid called pGlow, which has a lot of similarities to that plasmid, but has one major difference. So first, to point out the similarities, one similarity is the fact that it also contains the BLA gene, which is the selective marker for cells that are resistant to ampicillin. It also has an origin of replication, so that it is a self-contained piece of DNA. It also contains a multiple cloning site within which there are many restriction recognition sites, but those restriction recognition sites and the multiple cloning site are located not within the LAC-Z gene, but within a reporter gene called the green fluorescent protein gene. So rather than cells which will turn blue in the presence of XGAL, these are cells that will turn green in the presence of arabinose. So just so that you can connect those two ideas together. Before we move on to a different topic in biotechnology, I'd like to just show you really quickly how in vitro transformation is accomplished. DNA transformation is a process by which DNA can be transferred into bacteria. During rapid growth, the cell membrane of E. coli has hundreds of pores called adhesion zones. Incidentally, competent cells have more of these adhesion zones than normal E. coli, which is why we use them in our transformation experiments. The cell membrane itself is made up of lipid molecules that have a negatively charged phosphates, which presents a problem for us because the plasmid DNA, which we are trying to get the cells to bind and uptake, also has a negative charge. So we need to do something to negate that repelling that will be done between the cell membrane and the DNA. And what we do is add calcium chloride, because calcium chloride will ionize and the calcium will interact with the negative charges, creating an electrostatically neutral situation. Now that we've done that, we can move to our heat shock, right? Now that the charges have been neutralized, and we do that by first cooling down the whole experiment on ice in order to slow all the molecules down, and it stabilizes the membrane and it makes it easier to shield it. And then we do this very quick heat shock, and then that heat shock allows for the cells to uptake the DNA much more easily than they normally would. Moving off of the topic of transformation, but keeping with restriction enzymes, I'd like to talk just very, very briefly about one other really cool use for restriction enzymes. And that is that you can create what are called genomic libraries in order to study any organism on the planet. So you can create a library, which instead of having books, has individual clones that all have a small piece of the genome of an organism. For instance, the C. elegans here, you take the DNA, the total DNA from an organism, and you digest it with restriction enzymes. You take all of those fragments and you expose them to plasmids, which are also digested with the same restriction enzymes. So you get those same recombinant plasmids. You select for those recombinant plasmids, and then you keep every single one of them because each one of them has in it some portion of the genome of that organism, which can then be studied in order to figure out what that piece of DNA does and what role it serves in the organism that's being studied. We're going to return to that idea of a genetic library in just a moment using viruses instead of plasmids. But before we do that, I wanted to take a moment to talk about utilizing not a plasmid, but a virus as a vector. And this has its roots in the transduction that you learned about in the genetics lecture, where the transfer of DNA is mediated by a virus. So a virus picks up DNA from one cell, and because the virus is really just a DNA delivering packaging system, it will pick that DNA up and inject it into a recipient cell. And if that DNA can be inserted into the new cell's chromosome, then we will have a recombinant cell. One thing that in particular can be useful when you're dealing with viral vectors is that there are viruses whose natural MO is to inject their DNA into a cell and then integrate their DNA into the host cell chromosome. And one very commonly studied virus that does that are the adenoviruses. So adenoviruses have the machinery within them to cut the host chromosome and insert their viral DNA into the cell as part of the replicative process of that virus. So what they've done in vitro is taken this natural phenomenon and used it in order to utilize this virus as a vector, not of its own DNA, but of the gene of interest that you have put into this viral vector. So once the gene of interest is in the vector is allowed to infect the cell, but what it infects the cell with is not viral DNA. It infects the cell with the new DNA, inserts the new DNA into the host cell chromosome, and then creates a recombinant cell as a result of that process. And this has had some really amazing therapeutic success. It's not without its issues, but there have been some very successful gene therapy as a result of utilizing viral vectors to incorporate genes into a person who has a genetic disorder that results from their lacking a functional gene for a given phenotype. One success story that has to do with utilizing viral vectors is the gene therapy to functionally cure something called severe combined immunodeficiency syndrome. Severe combined immunodeficiency syndrome is an autosomal recessive disease, which you'll remember from general biology means that it can be masked by the presence of the dominant gene. And it is a disease that results from a mutation in one gene only, typically the adenosine deaminase gene, and that one gene deficiency causes a very low number of functional T cells to develop, a very low number of natural killer cells to develop, and it causes a person to have basically non-functional B cells, which we'll learn more about in our immune system unit, but basically means that this person has a completely non-functional immune system caused by one recessive gene, which is terrible, But the upside to it is that replacement of that one recessive gene with a dominant that can mask it will restore this person's immune system. So the normal gene is isolated from a healthy subject. That's our gene of interest. We make copies of that gene. We insert that gene into the viral vector. And then a bone marrow sample is taken from a patient who has this genetic defect. And that bone marrow sample is infected with this virus, which will inject the gene of interest, not viral DNA, into these bone marrow cells. And then those bone marrow cells are reinfused into the patient. And because they're bone marrow cells, which are a kind of stem cell for the rest of that patient's life, those bone marrow cells will create functional immune system cells and create for that person a functional immune system. Now, like I mentioned, there are some concerns with these gene therapies. Number one is that it's not useful in all instances where you have a genetic defect. They're only obviously useful when you have a recessive gene that's causing a defect. They're also only a useful therapy option when you can change stem cells in a person, and those stem cells can propagate the body in enough numbers to mask the natural cells in the person's body. And so this and certain cystic fibrosis therapies are the only real places where there have been a lot of success with this. There's also the concern if you have to give a person the adenovirus that you might cause an immune response. And there was actually a person who was receiving gene therapy for an unrelated genetic deficiency, and he unfortunately died as a result of a massive immune response. So all therapies, of course, are necessary to look at the pros and cons for, but just the idea that you can repair, in this case, a person's non-functional immune system, which would probably have led to their death as a very small child, is a really exciting proposition. So on the heels of that, the idea of utilizing a virus as a genomic library can be done very similarly to the way that we talked about it with housing genetics inside of bacterial cells. And this sometimes is a nicer way of doing that because it's sometimes a little easier to store plaques than it is bacteria. And it begins in the same way. You digest the genome of whatever organism you want to create a library for with a restriction enzyme. You build these phages that have this cellular genome fragment inside of them. You allow them to infect E. coli, and then you collect the phage progeny from each of those initial recombinant bacteriophages in the plaques that result in the bacterial lawn that you've grown. So you now have a library that consists of all of these bacteriophage that each house unique fragments from the genome of the organism of interest. As I mentioned, there is such a wide variety of biotechnological applications that we're only going to talk about a few of them. The first was restriction enzymes. The second is something called polymerase chain reaction. And the reason that we chose to look at these is because the restriction enzymes are actually enzymes isolated from bacteria, and the polymerase chain reaction is a way of cloning or making copies of DNA in vitro that relies on a DNA polymerase that has been isolated from an extremophile, which, as you know from previous lectures, is going to be a prokaryote. So, talking generally about DNA replication, we know that you have to do two things. Number one, you have to expose the nucleotide bases because they exist inside of that DNA molecule, and then you have to replicate the DNA. In vivo, in the cell, the normal process is that the genome serves as a template DNA. We know from Messelson and Stahl that the DNA molecule opens up and new nucleotides are added opposite their complements in the parental strand. So you need that template DNA, you need the nucleotides to add to the daughter strands of DNA, And you need, among other things, 3 very important enzymes. One is DNA helicase, which actually opens up and exposes the nucleotide bases of the DNA. You need that very important DNA-dependent RNA polymerase or DNA primase, which provides the 3' OH group to which the final enzyme DNA polymerase will attach and actually add the nucleotides to the growing fragment of DNA. So if you want to make this process happen in vitro, you've got to figure out a way to mimic these natural conditions. So first, you'll need a template piece of DNA, which can be any piece of DNA that you choose. You need nucleotides to create the new piece of DNA. In order to not have to use DNA helicase, you can instead use heat to denature the DNA to single strands. And you can use short primers instead of DNA primase in order to direct the synthesis of DNA to the portion of that genome that you would like to create copies of. And because you're using heat, you need to use a DNA polymerase that is thermostable. And there are 2 reasons that the DNA polymerase has to be thermostable. Number 1 is the fact that you have to get DNA to near boiling temperatures in order to denature it. The second is that you utilize heat in order to make sure that the primers bind only where they are annealed, to make sure that the primers bind only where they are complementary to the template DNA. Imagine you have the human genome, 3 billion base pairs, and you want to amplify or copy or clone the gene for insulin. You have to have a way of directing the DNA polymerase to that insulin gene, and you do that by providing primers. Primers are short, single-stranded pieces of DNA which are complementary to the outside edges of your gene of interest. So primers will allow you to direct the area of the chromosome that you're going to be replicating, because if they don't bind or anneal to the DNA, the 3' OH group will not be present, and the DNA polymerase, even if it's thermostable, will not be able to produce that complementary copy of that DNA strand. So during the PCR process, we're going to see that there are actually 3 temperatures involved. There's the denaturing temperature, which actually opens up the DNA molecule, and then there's an annealing temperature, and then there's an elongation temperature. The elongation temperature is the temperature at which DNA polymerase is active, which as a thermophile is usually around 75 degrees Celsius. So 75 degrees Celsius is enough heat that if those base pairs don't match, they won't anneal. As you see with the top example here, there's not enough complementarity between those 2 fragments of DNA, and so there's no annealing, and that means that there's no OH group there for the DNA polymerase to bind to. Opposed to the bottom example, which, because it's perfectly complementary to the template DNA, can withstand that 72 degrees Celsius. And so when DNA polymerase comes along, it will recognize that OH group and start to replicate the rest of that piece of DNA, utilizing that OH group to catalyze the reaction. So polymerase chain reaction has cycles that are made up of 3 temperature profiles. And each cycle begins with a denaturation step, which is about 95 degrees Celsius, which will denature the DNA into 2 single strands, exposing the nucleotides. Then it will drop down to about 50 degrees, maybe up to about 65 degrees to allow those primers to anneal. And then the temperature will increase to 72 degrees, which will test the fidelity of the primer template binding and activate DNA polymerase to copy the pieces of DNA only where those primers are bound. And so at the end of that cycle, you have exponential growth of the DNA molecule. You had 1, and now you have 2. And then you just keep cycling that process over and over and over again until you have billions of copies of DNA. In addition to needing that thermostable DNA polymerase, PCR needs a machine that can move this experiment through these temperature profiles quickly and precisely so that you can get as much DNA as possible. So a PCR reaction, you might imagine that you could do a PCR reaction on the lab bench. You could have a beaker full of boiling water, you could have a water bath set to 55 degrees, and you could have a water bath set to 72 degrees, and you could move that PCR tube through those three temperatures over and over and over again, and in about an hour you would have about 1 billion copies of DNA. Now that would be tedious, and of course there would be the time in between those water baths where it wouldn't be exposed to those temperatures. So today PCR reactions are done in what are called thermocyclers, which are machines that are set up to allow you to place your tubes, which contain all of the necessary ingredients for PCR. So they have the template DNA, they have the primers, they have the nucleotides, they have the thermostable DNA polymerase, and you put them into this machine, and this machine will move through the cycles of PCR. It will go from the denaturing step where double-stranded DNA is denatured to single-stranded DNA, and then move down to the annealing temperature where primers can bind to that single-stranded DNA, and then it will increase the temperature to the elongation temperature of 72 degrees so that DNA polymerase can catalyze the reaction of converting that single-stranded template DNA into double-stranded DNA. And these machines are really cool, both for that reason and for the second reason, which is you can set them up and they will run through all of these cycles for you. Notice here we have a setup in the thermocycler where we ask the machine to go to 95 degrees Celsius for 15 minutes. So we really make sure that all of our DNA is completely denatured. And then we have a second step where it stays at 95 degrees for 1 minute. It goes down to the annealing temperature for 1 minute at 60 degrees, and then it goes up to the elongation temperature of 72 degrees for 1 minute. And then you just tell the machine, do that step 2 through 4, 32 more times, and then go to 72 degrees for 10 minutes. So we get lots and lots of copies of that template DNA. And then go ahead and go to 4 degrees Celsius until I'm ready to pick up all of my newly created fragments of DNA. Again, I know you've heard me say this a couple of times. I'd like you to see this process happen dynamically, so take a moment to watch this video.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Once you have DNA molecules, whether you have created them and you just want to look at the entire molecule, or if you created them and you digested them with restriction enzymes and you want to look at them, you need a way of separating and visualizing those fragments based on their size, and that can be accomplished utilizing something called agarose gel electrophoresis, which utilizes electricity to move DNA samples through a thick gel matrix. And The electricity is used because DNA is a negatively charged molecule, so you can place a negative charge at the top of the tray and a positive charge at the bottom of the tray, and the DNA will be repelled by the negative and drawn toward the positive charge. Because you're making the DNA move through this gel matrix, larger pieces will be impeded as they try to move through the gel, so over time, you can create these banding patterns of DNA based on their size, and you can use those for multiple different applications. We're going to be looking at one of those applications in just a moment. We'll examine two such applications of visualized DNA, but first I want to show you what that process looks like in motion as DNA is drawn through an agarose gel with electricity. All right, pretty cool, right? So you can watch those molecules move through. All the largest ones are at the top, all the smallest ones are at the bottom. And once we have some idea of what those molecules of DNA are, we can actually use PCR in order to identify pathogenic genes. And that was done in this set of experiments here, where a bunch of patients were colonized by E. coli, and they were trying to figure out whether or not the E. coli that was colonizing these patients was actually the pathogen causing their disease. And it turns out that the difference between E. coli, the normal flora strain, and these pathogenic strains of E. coli is the presence of different pathogenic genes. So you can have primers that will anneal to the sides of these pathogenic genes and create copies of those pathogenic genes, but only in the strains which have those genes. So it's a way for you to look at the genotype in order to identify a phenotype in the patient. So on the very left-hand side, we have a negative control. This is just a PCR with no E. coli whatsoever. In what is labeled lane 3, we have an enteropathogenic E. coli strain. So you see one band there that indicates a pathogenic gene. Next to him in lane 4, you have an enterotoxigenic E. coli, which has 2 fragments of DNA that are associated with pathogenic genes. And then finally, enteroinvasive E. coli is shown in lane 5 with one other distinct and recognizable pathogenic gene. And then that is compared in lane 6 to E. coli, which are part of the normal flora. So all of these patients, if you did biochemical testing, would test positive for E. coli. So now we need to know whether these patients on the right-hand side of this agarose gel electrophoresis data are actually sick because of those E. coli strains. So patient 1 has a band that lines up perfectly with enteropathogenic E. coli, so that patient has this pathogenic strain of E. coli. Patient 2 and patient 3 have none of the genes that are associated with pathogenic E. coli. Patient 4 actually has a banding pattern that was not tested for in the initial experiment, but lines up with a 4th pathogenic strain of E. coli called verocytotoxigenic E. coli. Patient 5 does not have a pathogenic strain of E. coli. Patient 6 has the enteroinvasive E. coli, and patient 7 has enterotoxigenic E. coli. So PCR just of pathogenic genes can very quickly differentiate a normal strain of E. coli and pathogenic strains of E. coli, and it can do that for other disease-causing organisms as well, because many organisms cause disease only when particular genes that create toxins are present. Notice there was toxigenic, verotoxigenic, enterotoxigenic. These are all genes that produce toxins that are themselves the cause for the disease symptoms in the patient. One more way that PCR and restriction enzymes and agarose gel electrophoresis can be used biotechnologically is to create DNA fingerprints, which can be used to identify unknown fragments of DNA in what's called a restriction fragment length polymorphism. And restriction fragment length polymorphisms, I know is a large term, but if we break it apart, we have restriction fragments, so pieces of DNA which have at their end a restriction site, and they are of some certain length, and those lengths will vary based on where those restriction sites are. So you end up with polymorphisms, many shapes, many lengths, due to the presence or absence of these restriction sites. So here we have two fragments of DNA, one of which has that GAATTC recognition site for EcoR1 right in the middle of that fragment of DNA. So once you've amplified and you have billions of copies of that DNA, because they're all identical, they will all cut at that same site. And those two pieces of DNA will separate separately from one another on the agarose gel electrophoresis and be identified by these two bands that are shown on the bottom left-hand side of your screen. The other piece of DNA, because it's from a different organism, has a difference, a polymorphism in that site, and because that T has been replaced with a C, EcoR1 cannot recognize that as a restriction site, and it doesn't cut it. So now you get one large fragment, which, because all of those billions of fragments are exactly the same length, will all run together and create this one white line. These are DNA fingerprints. DNA fragment 1 from organism 1 on the left creates the fingerprint on the left, and the DNA fragment from organism 2 creates the fragment on the right. And that's something that we're going to utilize in lab as well. We're going to look at an experiment where PCR, restriction enzyme digest, and agarose gel electrophoresis will be used to determine the source of a salmonella epidemic. And that is all that we're going to talk about from the biotechnological side that has more to do with what we're doing in labs. What we're going to do before we wrap this lecture up, though, is talk about the industrial applications of microbiology. Now, as I mentioned, with the medical applications of microbiology, there are so many that we're not going to talk about all of them, but I would like to talk about just a couple of topics where microorganisms and the study of microbiology has had far-reaching effects in industrial or commercial applications. And that's because it actually is a subset of microbiology that is a result of the fact that microorganisms cause disease. And one of the ways that microorganisms very commonly cause disease is through something called oral fecal transmission. So whenever you contract something by eating or drinking contaminated water, you have been inoculated with what likely came at some point from the feces or stool of an infected person or animal. So that might be something that's on the hands of that person and then gets transmitted to their own mouths via auto-infection. It might be that that person's hands are contaminated and they touch a fomite, an inanimate object, which then touches your food like that plate. It can be that vectors like flies or other arthropods will land on the fecal material and transmit that to you. It can be transmitted through the soil. It can be transmitted in water. And there is lots of applied microbiology going to trying to stop oral fecal transmission, either because of toilet barriers or water barriers, and then what you can do yourself to protect you from these orally fecally transmitted diseases is hygiene and hand washing. So we're going to talk about this idea of oral fecal transmission much more in our last lecture unit, but here we're going to talk about how we can use microbiology to identify possible sources of oral fecal contamination of water. because lots of pathogens can be transmitted in drinking and recreational water. So that would mean water that you're swimming in. And one such test is something that we actually talked about when we talked about counting microbes, and that is the most probable number test, in which we can test water or food samples for bacterial growth by collecting a sample and then placing that sample into media in a series of dilutions in order to look for bacterial growth and the presence of gas as a result of bacterial growth. Once water samples are diluted, they're incubated, and after they're incubated, they're looked for the presence of turbidity and the presence of gas, and then those turbid tubes are converted to a code. In this case, we would have 3 positives in the first set, 2 positives in the second set, and no positives in the third set, which would give us an MPN code of 320, which you'll remember from previous lectures correlates to a most probable number of 93 cells per mil of water, right, plus or minus 18 to 420. So not only does this not give us a very precise number, it also simply tells us that microbes that are capable of fermenting a sugar called lactose are present in that sample. The question is that a pathogen, or is that just part of the normal flora of that environment? So among the things that we talked about in our microbial growth lecture, the other drawback to the most probable number test is that it only tells you that organisms are growing. And what you need to know in terms of microbial water pollution is there fecal organism growth there? And the way that we determine that is by looking for the presence of what are called indicator organisms. Indicator organisms, as their name suggests, indicate the presence of fecal contamination. And the best indicator organism that we have available is Escherichia coli, because it's ubiquitous in the guts of human beings and most other mammals, and it can't survive for very long outside of the body. So if you find E. coli, then you have found evidence that within maybe the last week or so, there has been fecal contamination of that area. And you might remember from the PCR reactions that I just showed you a few minutes ago that not all E. coli are pathogenic. So it doesn't mean that that E. coli is causing or is in danger of causing illness. It's an indicator that fecal contamination is present, and where fecal contamination is present and where E. coli are present, there's a very high likelihood that other more difficult to find or grow fecal contaminants are there. And in order to look for E. coli, you can do something called a mug coliform test, which is actually something that you will get to perform on a water sample of your choice. So the mug ColAlert test is a commercially available kit that has 2 reagents or substrates in it. One is ONPG, and you don't need to worry about what that stands for. The other is MUG, or MUG. And these 2 chemicals are important for 2 reasons. Number 1, they're capable of inducing 2 different bacterial operons. And the other important thing is that they change color in response to that induction. So the color change will indicate to you that organisms are present. Now, ONPG, which induces the lac operon, will induce that operon in any organism that has the lac operon, and that includes lots of organisms that are packed together into what we call the coliforms, the gram-negative bacilli. So getting ONPG, which is converted by beta-galactosidase, which is part of the lac operon, to turn to a yellow color, simply indicates to you that there are coliforms present, which, if you're talking about a recreational water sample, may be part of the normal flora. However, MUG, which induces the glucuronidase operon, will typically only be induced by E. coli, because this is a much less utilized operon. The production of beta-glucuronidase is indicative of the presence of E. coli, and so the conversion of MUG by beta-glucuronidase into this fluorescent compound, which you see in the photograph here is indicative of E. coli. And so if you were talking about drinking water, you wouldn't want to find a yellow color change, which would indicate coliforms, or a fluorescent color change, which would indicate E. coli, because drinking water should be free of microorganisms. If instead you're talking about recreational water, a lake, a stream, the ocean, something like that, coliforms would not necessarily be an indicator of anything dangerous in the environment, whereas fluorescence caused by E. coli would be an indicator of fecal contamination. So this allows you to be far more precise in what you're saying about a given water sample, not just something's growing, but something's growing that is a coliform, or something's growing that is E. coli. And when you have E. coli growing, you have very likely fecal contamination, and that should not be water that's being drunk or swam in until it's cleaned up. Continuing on this vein of illness as a result of contaminated food, we want to talk for a moment about food preservation, which we have talked about in previous lectures. When we talked about the control of microbial growth, we talked about causing osmotic stress to bacterial cells by drying food or salting food or sugaring food such that it causes osmotic pressure. All the water is drawn out of the cells and they're not able to grow on the food, which preserves it. We also talked about fermentation in previous lectures, where microorganisms will ferment, and oftentimes the products of that fermentation, the ferments, are noxious or toxic to the microorganisms. And so they preserve the food. And in addition, we talked about the fact that a lot of times, right, fermentation is a benefit not only because it preserves the food, but actually imparts different flavors to the food. So those are historic methods of food preservation. More recently, the idea of sterilizing and aseptic packaging, along with some of these newer technologies that we talk about in our control of microbial growth lecture, have led to foods that are sterile and have a shelf life of multiple years, right? And one common is canning, which can be done at home or can be done industrially, as is shown here, right? Food is cooked, it's put into a container, and that container is sterilized and sealed. And so those products can be stored at room temperature for years. That also can be done after foods are irradiated or exposed to high energy electrons or high pressure. Any of those things will sterilize the food, and then it can be aseptically packaged into sterile containers in order to preserve the foods for much, much longer than they would be naturally safe to consume. As we mentioned, this is not a new concept, and in fact, Louis Pasteur came up with something that he called pasteurization in the 1800s, where he was actually heating wine at 60 degrees in order to kill pathogenic bacteria and deactivate spoilage enzymes. And when he published those works, as you can see here in 1876, that was picked up by Franz von Soxhlet, who applied this idea of pasteurization to milk and came up with this modern idea of pasteurization, where milk is exposed for 20 minutes to 60 degrees Celsius in order to reduce the number of spoilage organisms. Now, milk exposed to historic pasteurization still has to be refrigerated because it still has organisms in it. It's not sterile, but it will last for much longer than it would otherwise. Today, our milk is exposed to what we learned in our earlier unit. It's called ultra-high temperature pasteurization, exposure to 135 degrees Celsius for 2 to 5 seconds, and that allows the milk to be pasteurized, but to maintain more of its natural flavors. And this method of pasteurization, though it doesn't sterilize the food, eliminates Listeria and Salmonella, Staph aureus, Yersinia, Campylobacter species, enterohemorrhagic E. coli strains, and so it prevents a lot of disease transmission in addition to the fact that it provides a longer shelf life. Now, one drawback might be that it also eliminates volatile aromatics, which can actually change the taste of foods, and so there are some things that people prefer not be pasteurized so that those volatile, meaning chemicals that would evaporate at these temperatures, are retained inside of the food item. One of the most well-known fermented foods is beer, and it's also one of the oldest. In fact, the history of brewing grains in order to create a fermented beverage is 13,000 years old. We have archaeological evidence shown at the bottom of the slide here of brewing residue that was found in Israel. We have beer stone residue on jars from 5200 BC. We actually have a beer recipe recorded in hieroglyphs from 2000 BC. In 1800 BC, beer is mentioned in the Epic of Gilgamesh. Not only is beer mentioned, but it's actually attributed for why Gilgamesh's sidekick Enkidu actually agrees to go on his missions with him. He gets drunk and decides why not. In the very famous Code of Hammurabi from 1754 BC, not only do we have acknowledgement that beer was being made, there are actually infractions and punishments listed in the Code of Hammurabi that forbid brewers or merchants from watering down the beer that they served, and had they been caught, they would have been forcibly drowned in the watered down beer that they were trying to pass off. In addition to that, in these cups and jars found in northern Iraq in 500 BC, we have beer residue. So beer is an incredibly old and actually very useful fermented food. Early on, it was used when water, safe potable water, could not be maintained. People would ferment the grains inside of the water and create these fermented products, which could be drunk. Now, beer is not the only microorganism associated food product. Anytime you eat cheese, you are eating something that was fermented either by lactic acid producers, which produce sharp cheeses. If you're eating soft cheeses, you're eating something that's been acted on by molds. And microbes are actually responsible for the enzymes which create cheese, which may sound yucky to you, but the option prior to genetically modified organisms making this rennin, they actually had to take a piece of the stomach from calves or other baby animals who were drinking milk from their mothers in order to make cheeses. Of course, breads are made from yeast fermentation, as are alcoholic beverages, right, which includes wines, the beers we just mentioned, and rather interestingly, the production of rum, which was a chief component in grog, which was handed out daily in the Imperial Navy as a way to provide clean water and provide vitamin C to sailors so that they would avoid scurvy. There was a proprietary rum that was produced by the British Navy, which you can purchase now because they don't give it to guys on ships anymore, called Pusser's rum. And it was a 1-to-4 ratio of rum water to lemon juice and sugar. And that became what's known amongst sailors throughout the world as grog. And it was initially given to sailors in order to provide safe water and a source of vitamin C so that they wouldn't get scurvy. Moving on to less appetizing options, there are two other industrial processes that are heavily reliant on microorganisms, and that is copper ore extraction, which is accomplished by Thiobacillus ferrooxidans, working to oxidize iron so that it can be used again to leach copper out of the earth, and multiple kinds of microorganisms are utilized in the treatment of wastewater before that wastewater is returned to the environment. In addition, the same recombinant DNA technology that we talked about at the beginning of this lecture can be used not for pharmaceutical products, but for industrial products, where genes of interest from some industrially useful organism are integrated into bacteria through the process of transformation, and then the genes from those can be inserted into plants. They can be used for bioremediation, they can be used for other industrial applications, and they can be used to treat patients. So what we're going to do is just take a quick look at some of the industrial applications that have been utilized for a wide variety of applications. We'll start with laundry, which was helped along in the early 1900s by the use of enzymes, and still happens today. You go to the grocery store and look, and you'll see all of these enzyme actions in your laundry detergent, and that is thanks first to a chemist named Dr. Otto Röhm, who was using enzymes that he had extracted from cow stomachs for bleaching leather, and his wife speculated that they might also be able to remove laundry stains, and that sparked some interest in Otto Röhm, and in 1914 he patented the first enzymatic detergent. Fast forward about 100 years, and tons of microbial proteases are used in order to create more of a green chemistry. They range from biofilm remediation to silk degumming to fibrin clot dissolution to keratin residue processing, which may seem super crazy, but 8.5 billion tons of poultry feathers are produced annually. It used to be that a very high temperature and pressure process was required. Now they add keratinases, which chew that up into proteins, which can then be utilized as protein supplements. These industrial proteases are used in leather preparation. They're used in wool biopolishing. They're used in X-ray technology to recycle the silver from the gelatin and the emulsion layer so that that silver can be reused. And they're even used in contact lens cleansing solutions. The enzymes in the contact lenses digest biofilms and prevent the attachment of pathogens to the contact lenses and make them safer for use. These industrial processes, which utilize these microbial proteases, produce about $4.2 billion annually. And they go from things that you might add to your foods, like xanthan gum, which is very popular amongst people who are gluten intolerant because it adds a sponginess back to breads and other things when gluten can't be utilized. As you can see from the picture here, it also creates a much smoother consistency in materials. So here you have one made with xanthan gum, one without, and so the xanthan gum creates this creamier, more appetizing look to the product. Xanthan gum is found all over the place. It's found in hot sauces, ice creams, and it's produced by bacteria of the genus Xanthomonas. Microorganisms are also used to produce high fructose corn syrup, and there are about 4 million tons of that produced annually. Microorganisms are used to produce citric acid, which has an especially interesting story behind it because of its connection to a very important pharmaceutical product, and that is penicillin. And penicillin would not be around if the work of James Curry at Pfizer was not done previous to that in order to produce citric acid from a mold called Aspergillus niger. And he did that in these giant containers, which are going to become very important to the story of penicillin. This citric acid, though, is important in and of itself. About 2 million tons are produced annually. About 50% of those are used as an acidity regulator in beverages like Coca-Cola and Pepsi and things like that, 20% in other food applications, 20% in detergent applications, and then 10% for pharmaceuticals and cosmetics and things like that. So important on its own, but also, as I mentioned, because of its relationship to penicillin. And the relationship of citric acid to penicillin is James Curry, who worked along with a 16-year-old assistant named Jasper Kane to up the production of penicillin after its discovery in the 1920s so that it could be used in order to protect Allied troops during World War II. And the problem with penicillin is it's finicky, it's difficult to grow, and even when you produce penicillin, there's about a 1 to 10,000 ratio of penicillin to the broth. So you have to have a ton of broth, a large-scale production, in order to make enough penicillin to be therapeutically useful. But if you have these giant vats, the problem is that this mold is an aerobe, and the bottom of the vat would not support the growth of that aerobe. So Curry and Kane devised 14 75,000-gallon tanks, and they used them in 1943 to start producing penicillin. And it was really exciting because right off the bat, they produced 5 times the penicillin that they thought they would be able to. And on D-Day, every Allied troop had a dose of penicillin, and 90% of those penicillin doses were made by Pfizer through the work of these two men, James Curry and Jasper Kane. And that work was done utilizing the deep tank fermentation technology that Curry had started working with in Pfizer to make citric acid. And these deep tank fermenters, or bioreactors as they're sometimes called, are basically enclosed containers where every conceivable aspect of the environment is controlled. If you get too acidic, a base will automatically get added to raise the pH. If it gets too alkaline, an acid will be added to lower the pH. If it gets too warm, then a cooling agent will be added to the outside to cool it down. If it gets too cool, it'll get heated up. It's aerated, and it allows for the organisms inside it to grow to stationary phase, and instead of moving into death phase because of the buildup of toxins or the lack of nutrients or the lack of space, those organisms are capable of staying in stationary phase, and that allows for a maximal amount of whatever you're looking for in this fermentation product to be harvested. So when we're talking about deep tank fermentation in bioreactors, notice we're not talking about lag and exponential and stationary and death phase. What we want to talk about is cells that are at a low concentration and cells that are at a high concentration. And so those are differentiated by trophophase and idiophase. So trophophase is cells anywhere before stationary phase. Idiophase is where the cells enter stationary phase. And the reason why this idiophase idea is so important is because many things will not be produced unless the cells are at high concentration, which may sound familiar to you, right? That's quorum sensing. So for instance, penicillin. Organisms produce penicillin to kill off anybody that's competing with space for them. So they don't need to produce penicillin until it gets crowded. So they use those interspecies chemical cues and intraspecies chemical cues from around them. And when those chemical cues get to a high enough concentration, they realize that the cell density is high enough that they need to start killing off the competition. And so they produce penicillin only during idiophase. So that means that if we want to harvest penicillin, we need to get the cells to idiophase and keep the cells at idiophase for as long as possible so that we can extract as much penicillin as possible. Now, even if you have something that's produced all the time, say ethanol, which is another commercially useful product, you still have a vested interest in getting the cells to idiophase because the more cells there are, the more ethanol they will produce. So whether you are producing something that is what's known as a primary metabolite, something that is produced no matter where the cells are in this cycle of growth, or a secondary metabolite that is not produced until the cells enter the idiophase portion of growth, you will want to have them growing in deep take fermentation bioreactors so that you get the maximal product necessary. Now, as I mentioned, the industrial products, deep tank fermentation and other things are so varied that it really would be difficult to mention all of them. But just for instance, amino acids are produced, proteins are produced, vitamins, steroids, antibiotics, as we just saw, drugs, ethanol, as we just saw, methane gas, all sorts of different things. And they're all made because bacteria have been found that do those things naturally. And then that natural ability has been exploited in vitro in order to accomplish a particular task, which is either pharmaceutically or industrially important. And one such organism is E. coli, who has been bioengineered to do all sorts of amazing things. And I just want to highlight two areas of recent research, one being done by DuPont Industries in London, where they have E. coli biomanufacturing 1,3-propanediol, which is found in composites, adhesives, laminates, coatings, moldings, carpet, and clothing fibers. There are about 140 million pounds made annually, and by biomanufacturing it using E. coli, you use about 40% less energy than traditional methods. Another is the University of Toronto, which has been biomanufacturing nylon precursors utilizing E. coli, which isn't so much a benefit because of the energy that it saves, but because of the toxic waste that is typically produced during the production of nylon precursors. Conventional production uses petroleum-derived benzene and releases nitrous oxide into the environment, which you might remember from previous lecture is a carcinogen. So just a little shout out there to our friend E. coli. As we learned earlier, there are pathogenic strains of E. coli, but he really is sort of the microbial version of the guinea pig. So 3 cheers for E. coli.